Stanford University.
Good afternoon. My name is Dennis Allison, and together with Andy Freeman, we organized this class, which is EE 380. It's a colloquium which presents a broad range of topics, ostensibly tied in some fashion to computer systems. It's the descendant of the Digital Systems Colloquium and the Computer Systems Lab Colloquium, but now identified as a EE class. It's open to anyone who would like to attend. It's run as an open lecture. It is streamed on the Sitten network and available from our website, http://ee380.stanford.edu. The class is available as a one-unit course, satisfactory or unsatisfactory, for which you can sign up, and you can do this every quarter that you're enrolled at Stanford. The quid pro quo is that if you take it as a course, you have to write a short, pithy comment about each of the lectures and fill out a evaluation form at the end of the quarter. That's about 5 minutes work each day for each class at most, and nearly everyone is successful at completing it. If you don't, we will pursue you to the ends of the earth and make you do it. We don't give incompletes. We encourage people to attend the class live because the interesting stuff happens off camera in general. On the other hand, a substantial fraction of the audience does choose to attend by video. We suspect that people tend to try to batch all of the videos in a single day and complete it just before the deadline. That is acceptable but not encouraged. The average, the deadline normally is the last day of finals, and it typically gets moved around a little bit when the calendar makes graduation and the last day of finals and everything sort of a harmonic convergence. But nonetheless, we do like people to attend and ask questions. The course material tends to be roughly graduate level, but the variation in technical level is fairly high. We let interest to trump technical level from time to time, and you can discover by looking at the program for this coming quarter. We're always looking for new speakers and suggestions for topics, and we have at this point a couple of open slots for the quarter, and of course there's 30 slots next academic year. One of the nice things about having Andy introduce as opposed to me is that Andy tends to think through and write really quite lovely introductions. I don't do that. I just sort of toss the speaker out in front of you and see who wins. The speaker for today is Drew, MD. He's in bioengineering, a Stanford department, and does extremely interesting things with biological components. And so with that, let's welcome Drew, MD.
My lab's over in the Y2E2 building in the quad that's coming up. We're right below Coupa Cafe, and the lab's under negative pressure. So the work we do goes through the ventilation system, yeah. So I wanted to start with a research project that might relate. I want to get to 8 bits, 1 byte of data storage. The trick is I want this to operate inside living systems. And so how could I engineer 8 bits of programmable data storage that operates, for example, inside every cell of your body? Yeah. So this is a project we've worked on for about the last 3.5 years over in Y2E2. The specification in terms of performance requirements are, again, it needs to operate inside living systems, needs to interface with biochemical functions. It probably needs to be stable across evolutionary timescales. I might want to do things like count cell division events or aging processes. It doesn't have to be fast. It could be slow. It could run on millihertz frequency. That'd be great. If I could update the state once every tens of minutes, no problem. Right? If I think about the application, there are going to be things like cell division events again, differentiation from an egg to an adult organism. It's all sorts of weird, different things. 8 bits turns out to be quite a lot for engineered biological data storage. The first BioBit was demonstrated circa 1985 with cross-regulating transcription factors. And the state-of-the-art this year is a one-shot count to 3 thermometer code counter. So 1.5 bits, maybe. But it overcounts if your input pulse is too long. So we set out about 3 years ago to really dig into this challenge of engineering scalable genetic data storage systems and see where that would take us. I have to admit our motivation was really to confront fundamental engineering problems. Composition of biochemical elements, reliability of performance in the context where you've got thermal noise inside cells, and also evolutionary processes and how you could store state when you have mutations showing up. I only need, though, in the end of the day, this is going to come back to haunt me, I hope, 8 bits. 8 bits is quite a lot of data storage for living systems. The number of times a cell might divide before it comes senescence stops dividing is typically a few hundred. So if I get 2 to the 8th, that's going to give me a lot. The differentiation events in an organismal lineage might be a few dozen to a few hundred. So I could record everything. And so I don't need gigs. I need 1 bit to start. And that's about an order of magnitude beyond the state of the art. And the state of the art has not progressed very much since 1985. That's the context as a rough pass.
Why didn't you just use DNA?
Well, that's what we're going to do. The trick is, no, it's a perfect question. Why not use DNA? The trick is we don't have really good tools for writing to DNA inside cells. So what I need to be able to do if I want to store information into DNA, which is a wonderful natural material for storing information, is I need to create tools that allow me to write state to DNA and hopefully rewrite. All right, so
be careful they're not abused.
Could be an issue.
You have to have code separation.
Yes.
These are all good problems to have. I'm just going to tell you a story about getting to 1 bit that we like. All right. This is how we do it in an abstract representation. The horizontal line is meant to represent double-stranded DNA on a chromosome. And then we have an engineered register, which internally we called the flippy. And this is a specific sequence of DNA. It's a couple hundred nucleotides or bases long. And what we're going to try and do is take this piece of DNA inside a chromosome of a live cell and cause it to be flipped between specific sites. And then we'd like to cause it to be flipped back. And if we could get flip and flip back, we'd have set and reset, we'd have writing and what have you. And this would be our unit memory element, or whatever the right word is for all of you. Right? This would be our register.
So how would you read it?
Aha. Because we can flip DNA, the thing inside the flippy can be a genetic element. So it can have function inside the cell. An example of a simple function, which we're going to use here, would be called a promoter. A promoter is a genetic element that initiates transcription, the reading out of DNA to make mRNA and eventually maybe proteins. So if I put a promoter, call it that arrow inside there, I can control the orientation of that arrow. So double-stranded DNA will have a directionality, and I can have that arrow, meaning that promoter, activate gene expression to the right or the left. So for example, if I put a red gene, a gene encoding rather a red protein to the left, and I set this in the right to left orientation, my cells might turn red. And I could declare that to be a 1 in some code. And if I reset it back to green, the red gene no longer is expressed, the green gene is expressed, the cells turn green. Call that a 0. So to do this, we need some flippers. The flipper we're using here is a protein adapted from a virus. It's called an integrase. The natural function of an integrase is the following. When a virus infects a cell, sometimes viruses go dormant by integrating their genome into the host chromosome. The enzyme that mediates that reaction, that integration reaction, is called creatively the integrase. It was determined decades ago that you could change the sites that the integrase recognizes, and instead of catalyzing an integration reaction, if you had the sites in the right orientation, you could instead get a flip. So we're going to take advantage of that. In other words, all we need to do to get the first setting reaction to go is give a little pulse of expression of this integrase. Hopefully, we'll get this flipping. So let me show you how this works. This is work by Jérôme Bonnet, a postdoc from Montpellier, France, and Pakpum Subsutorn, a graduate student in BioE from Thailand. These are cells, these are E. coli cells. We're working with E. coli because they're fast to grow and they're safe in the lab. They've been defanged relative to pathogenic strains. Although the enzymes we're using are taken from a context such that they're known to work in any organism people have tried. So they work across kingdoms. So we like the setup we're developing here in part because the biochemistry we're using should work in mammals and plants and you name it. But we're showing, everything I'll show you here is with bacteria. So this is a microscopic image of E. coli. They're green. This represents the 0 state. This is a 2-color plot from a cytometer that measures the color of thousands, tens of thousands of individual cells. If you see a splotch here, this is a heat map, the frequency of cells coming through this machine that are in, high on the green axis and low on the red axis. So green on the X, red on the Y. We hit these cells with a pulse of a set signal, which leads to production of the integrase, we hope. And you can see that the cells switch state. They become red. The cytometry blob moves over here, low green, high red. Call that a 1. And importantly, when we remove the set signal, this input, the cells stay in this state. And I just want to restate this from the perspective of, say, an SR latch, where you might have 2 coupled NORs. Right? We don't have that back wire in this system. Right? So this is a passively stable system. It's not bistable in any sense. But it turns out to be the case, much like your original question. If you can write to DNA and get your information in there, that might be really tasty. And it turns out we've tested this. This thing will hold state in the absence of any inputs or bistability for over 100 generations, 100 replications. That's as far as we've tested. Yes, sir?
Are you going to talk about time?
Yeah. This input pulse is about 10 generations long in this experiment.
What's the time constant for a generation?
120 minutes. I'll show you how fast we can make it go at the end. We need to get the input pulse down to about 30 minutes, right? Or half a cell doubling, if we want to do things like count cell division events. That's what we're aiming for. But this is an experiment that's about 3 years old. The trick in getting this to work turns out to be the following. You need to have a control over the expression of this one gene such that you don't have leaky expression in the absence of an input signal triggering a spontaneous event. And then you have to have enough expression to trigger switching with high reliability within whatever your window is. And that turns out to be really non-trivial, as I'll get to if we have time. Just that engineering of a few molecular components to get that switching behavior.
Parity bit?
Dude, we've got 1 bit. Just come on.
So you have to start with 2. Yeah, parity.
Welcome to bioengineering.
I proposed that some time ago.
All right, so I'm going to claim this works within the conversation we've had so far. How do we get a reset? We're going to take the same enzyme and we're going to modify its activity by co-producing another protein called excisionase, which is supposed to reverse the directionality of the reaction. In natural systems, if you have a virus genome integrated into the host chromosome and you make both these proteins, that'll cause excision, popping out of the virus genome and reinitiation of infection. And so that's what this is supposed to do. Now, for context, people had, a couple years ago, gotten flipping in one direction working really nice for this one-shot thermometer code counter. And what we've been focused on is getting set and reset working together in the same cell so we could begin to imagine getting to combinatorial data structures. Right? So if you had n registers, you could count to 2 to the n, as opposed to just n. So we've spent a lot of time on getting this working, and then getting this working, as you'll see, in the context of a set. So this is what cells look like. They're in the red state. We give a pulse, what I say, 1,200-minute pulse of protein expression. The cells all go green. We remove this pulse. I don't know if you can see it. They mostly stay green. See, this one cell here is red. It reverted back. It corrupted during the relaxation from the reset input. But we're pretty happy with this. Mostly works. Now here's where it got really crazy. So that last slide was about 3 years ago. The next 3 years are spent trying to figure out how to take the set and reset and get them working together in the same cell. And let me just give you a taste of the types of problems we ran into. For example, in order to get this to happen, you need to have a lot of the excisionase. These are self-mixing molecules bumping around. And so if you don't have a driving stoichiometry for the excisionase, it might be that the integrase finds itself alone for a moment. And when you think you have a reset, whoop, it goes back and sets. Right? Another thing that could happen is, following a reset pulse, we have to start to worry about how these proteins are being processed or degraded, recycled by the cell. If, for example, as you can imagine, the excisionase goes away first, following a successful reset, now all of a sudden we've just established the conditions for a set, and we're going to corrupt the reset. Now we fix this by doing things like more highly expressing the factor, the excisionase protein, stabilizing the protein so it's not degraded or recycled, and that works great. But guess what happens? When you put the gene cassette for having really strong expression of excisionase that's rock stable, it gets sputtered, it gets read out in a spontaneous fashion independent of any input signal, which means that when you try and operate a set function, the cell already has the excisionase protein hanging out because it's just spontaneously expressed above some basal level that we can't clean up. And so that corrupts the set and turns your set operation into a bidirectional set-reset cycle. How do you fix this? You destabilize the excisionase, you reduce the level of expression, which, oh, by the way, gets you right back into the other regime. Right? Now, I'm saying this like it's self-evident, but to be honest, we didn't understand this very well. And so we sort of came to this understanding empirically over a period of time. Eventually, we come up with a mathematical representation of this. So we sort of parameterize, for example, the degradation rates of, on the X, the excisionase, on the Y, the integrase proteins, and then can predict what we would expect the reliability or efficiency at a single cell level of a switching event would be. So regions where there is a red zone is where the set or the reset would be expected to work in parameter space. So for example, we believe that we were operating reliable sets at this point, but you then come over to the reset space and you get something that is not clearly 0 or 1, but now in this intermediate state, bouncing between. We fix this by stabilizing the excisionase, for example, that cleans up our reset, but that then in turn corrupts the set. Right? You can see where this is going. This is not crazy complicated. We would like to be over here. Right? If you take the two functions together and figure out where it is in the parameter space you'd like to be operating, this top right island, that's the kinetic parameter spot where you get set and reset working. And then you see this title here. This title says, Precise Engineering of Central Dogma Needed. Central dogma is the name in biology for DNA makes RNA makes proteins, and then RNA and proteins get degraded. And precise engineering means we would like to be able to forward engineer genetic elements to realize precision engineering of the reading out of DNA and RNA and the processing of mRNA and proteins. To be very specific, if you look at the scale there, that's a base 10 logarithmic scale. So that island is about a fourfold range that we want to be hitting. I won't get into it here, but the size of that island gets smaller when you reduce the cycle time, when you try and speed things up. Right, so as we're speeding this up, we're trying to hit a smaller and smaller island. And ultimately, right now, we care about forward engineering pieces of DNA that get molecules to twofold concentration ranges in cytoplasm. This turns out to be ridiculously hard to do. Why? Because we're working with found objects, meaning the control elements we use to do genetic engineering today have been harvested from nature. They're quite literally found objects. They have not been reworked to support reliable composition and precision control over the central dogma. I'll come back to that. Thus, 3 years and 750 design, build, test cycles later, we have the following artifact. And again, this is really mostly Jerome Bonnet's work. And I consider it to be heroic, not just in a self-interested sense, but it really is heroic labor and ingenuity. We have a set and reset input signal. These are generic transcriptional signals. The set is the integrase coding sequence. The reset is the oxygenase integrase coding sequence. This is a work of art. It's a handcrafted work of art. So for example, the start codon for the integrase gene, if you study the classical genetic code, you'll know a start codon. All genes begin with ATG. Not these. These begin with GTG, which we found we needed to use to get the right level of synthesis. You'll see that in nature, but typically not in genetic engineering, and so on and so forth. So there's a lot here. I'm happy to give you a paper if you'd like to read how we made this and get into the details of it. And this is what the operation. So now we're just looking at the fluorescent signal in the 0 state. Okay, so we start off with cells in a 1 position. And then the fluorescent signal when reset comes back up for 0. This is the reset pulse. Again, 10 cell doublings, 1200 minutes. Actually, is it? Yeah, that's right, 20 hours. Okay, then the reset signal goes away. The cells hold state. We set them, they hold state. And hopefully this will go. And then we can do that basically as many times as we want, so long as many times as we want is less than 10. All right? You get a sense of what this looks like. So what is this? 1, 2, 3, 4, 5, 6, 7. Right, that's about how many times we've driven this. But it looks pretty good in that you can always clean it up and restore to a pretty good 1 state with a set. Right. This bottom series is with shorter input times, about 2 cell divisions. So a 4-hour input pulse, which means we need to get down about another factor 4 in terms of the speed. Speed it up about.
To make it stable, or to clean up, quote unquote.
Well, that's.
How many cycles you have to make? Wait, to converge into a good state.
Just about 4 hours. And in fact, to be honest, we really think we're going to be able to get this to 30 minutes, right? So I think we're on a path where we're going to get this working.
What happens after 10? Have you just not done it, or
Yeah, that's right. I mean, this basically is an expert working in the laboratory doing serial manual passaging of cells for a week, right? And then each of these splobs is going to the cytometry core in a different building and collecting the data and then processing the data. You know, so it's expensive to collect the data. But I'm not showing you data where, for example, the stability of the register has been established for 100 doublings. Right, so that's separate experimental data. Again, if anybody wants the paper for this, we're really excited about this result. I'm comfortable saying this is probably the best genetically encoded data storage element on the planet right now.
You
All right.
hacked
DNA of the E. coli?
That's right. This data registers on the chromosome of the cell.
Remind us of what the DNA of E. coli looked like. It's a prokaryote.
It's a prokaryote. It's a circular piece of DNA about 4.65 million bases long. And we're taking over about a kilobase. Pop quiz. Why is this so hard? Genetic engineering was invented here at Stanford in part a generation ago. How come I'm showing up now in a new department of bioengineering and it takes me or my team of order 1,000 attempts in order to get a 3-gene system working? Could be. We're going to make this pop quiz real easy. I'll make it multiple choice. Very few people have been working on getting better for the last generation. Business or politics is selecting for something different than getting better. It's impossible. It's really complicated. It's very different than anything we've seen before or other. I think they are. Could be all of the above.
Oh, it could also be other. Have you seen the talk by Mara Prentice from Harvard?
I don't know.
You should have, because the first thing she says, she's an experimental physicist. So she's talking about DNA. The first thing she says, biologists have it all wrong. It doesn't work the way they claim it works. And then she gave her explanation.
Okay.
So maybe you should talk to her.
Okay.
Is her
Thank you.
explanation correct?
I'm sorry?
Is her explanation correct?
Yes, she has enough data to prove it. It's actually, it's an interaction, not just between the double strand, but the single strand and double strand. So it's a three-way interaction. Which as an electrical engineer, we like that. We have 3 ports.
You want a 3-terminal device.
Yeah.
Yeah.
That makes it more stable in a certain sense. That's part of the reason I ask you more than one code. Have you looked at, like if you take 2 codes, either a repetition, and you know it can spiral, or if you do it like a dipole against each other.
I think you bring up.
Those
I
seem to
think
be more stable.
I think you bring up a good point. You know, one of the heroes of engineering for me that I've learned from is Tom Knight, and he always, when working with biology, was quick to bring to the conversation, what's your reference frame? How are you thinking about encoding signals in state? And I'd be very comfortable if somebody showed up and said everything I'm using right now as the principles of the system are naive, and we could have a much more sophisticated coding.
Tom Knight could be wrong too.
He could be wrong too, but all I'm telling you right now is, so I'm very interested in that. I think those are very valid points of departure.
Arrangements, and there may be more
Yeah.
dipoles or quadrupole, whatever.
My approach tends to be, at this point in my life, where if I'm lucky I've got another 30 or 40 years to crack at this, if I'm lucky, to be very practical in an empirical sense, and to just try stuff and see how far we can push it, and when things break down, then figure out a different way to go about it. So I didn't know if we could just reset, right? And we started with that. I want to come back to this though and share anecdotes, and I think you're insightful in that I would agree that A and B are related. For example, one of the great things to do in engineering is to borrow ideas when you're doing something. So there are textbooks on bioengineering, and since we're starting a new bioengineering program here, might as well read the textbooks and figure out what to teach, right? So if you get this book and you read it, in the preface, sentence 3, you'll find the following. As a declarative non sequitur. So, okay, what this I submit as evidence for, but there's a lot of other experience. Although many great engineers have been working with biology, most engineers and most engineering factions, communities, have not engaged with biology, have not really gotten into it. And we suffer from that. And it's one of the reasons I instantly agreed to come here.
And vice versa.
Yes, I would agree. Yeah.
So if they believe in that sentence, is the book any good?
No. Another weird experience. I was testifying before the Energy and Commerce Committee in the House a little while ago, and we got asked whether or not could we engineer a genome that would predispose folks to vote Republican. Now, it's a funny question, but it's also a good question, because the answer is yes. We've got to have some conversation about that. But I submit this for consideration in the sense that biotechnology, when it does get engaged, tends to be focused on problems that are human problems of some pressing nature. Please cure my disease. Save the environment. And as a result, the teams that get assembled are on these focused missions that make it very hard for them to take even a half step back and ask and then work on, how could I get better at doing this type of work? In other words, I believe that the first generation of genetic engineering-enabled biotechnology has been overdriven by applications, that we've not managed to sustain incremental improvements in tools. I believe this is still true today. When will the drop-in biodiesel be ready? What disease am I curing? What is the one application of genetically encoded data storage? That's my experience. And it's my experience as an advisor of basically many leading biotechnology teams. Okay, so I want to zoom out and go meta. This is a dash of Lynn Conway. All right. So it has been the case that for other types of material, we've gotten better at engineering it. And I want to walk through a case that I'm more familiar with because I'm trained as a civil engineer, and then a case that's very risky for me to bring forward because you're all expert in it. But let me get to the first one first. So can we make stone easy to engineer? Start with this, build artifacts from these things, run into limits of what you can build. So what do? Some people become incredibly excited about the differences and they go study the rockiome. They become geologists and characterize all the rocks on the planet. That's great. But other people become fed up with these artifacts, so they do this, among other things. They begin to invest energy to regularize the material. And what's very cool is as soon as you make simpler elements, you can make more complicated artifacts. It allows for standardization that then leads to coordination of labor across time and place, accrual of improbable value that would otherwise be impossible to realize. Also, this happens. People become fed up with rocks, even when they're standardized. So we make synthetic rock. Or if you walk around the quad here at Stanford and you look down, you'll see this stamp on the paving around the perimeter of the old quad, artificial stone. It was a point of pride that Stanford had managed to implement artificial stone around our original quad because we were fed up with, I don't know what, cobblestone or something. And when you do this and make many other advances in terms of process engineering, you make still more improbable artifacts. All right. So what do I want to steal from our Stone Age as a process engineer? Invest energy to regularize material. That could be a cool thing to try out. Improve physical assembly and functional composition. Develop standards that allow for coordination of labor across time and place. How do I get somebody in Melbourne, Australia helping me make data registers? Going to need a lot of diverse tools, not just on the material, but on design and construction and testing. There's probably more, but I have to admit, a large part of my business is to scour the history books of engineering and try and identify things we can steal and just try out. All right, now here's where I'm on thin ice. What about this stuff, electronics? Well, I've seen pictures of what computers used to look like. The killer apps were weapons design. Not so compelling, but relevant in the context of people freaking out about biosecurity today, which we can come back to. Less than a human generation goes by and artifacts like this show up. And then another generation goes by and artifacts like this show up. How can I explain this? As the civil engineer, now the bioengineer. Well, I can find photographs from, I think, the 1970s. These are very interesting photographs. These photographs must show something very important in the process of engineering computers. I don't know what it could be, but what I note in these photographs is I can see the human beings. This is an interesting time because this is when genetic engineering is invented. Right? So just make that connection for a second. Now today, I have a hard time finding those sorts of photographs. Instead, when I ask people to explain to me what is it that I might be seeing in these old photographs, they say, oh, you should go see Cadence Design Systems or Synopsys or something like this. If I wanted to go see genetic engineering today, though, I'd go see people and manual labor. And as I start to dig into this, I learn about all sorts of things I take for granted, meaning I've never thought about before. Metrology. If you talk to my colleagues and they hear the word metrology, they'll typically respond, meteorology? Information theory and devices. That's pretty amazing. Programming languages. Standards. The ohm. Jerry Sussman taught me that the ohm was a person with a beard. You are so lucky. You have a unit of measurement. I don't have a unit of measurement for transcription. Silicon wafer fabrication. That's pretty nice. Computer design, EDA, control, When I go buy a computing machine, I don't think about any of this. But there it is. And I think this probably is secret to explaining from 1950 circa to 2012. So what meta lesson do I want to steal from you? Instead of thinking about the applications, I need to think about the tools, and I need to figure out how to sustain incremental improvements in the process tools so I might have a chance of realizing geometric improvements in the complexity or quality of the artifacts I can make, or others can make. I don't know if that's right or not, but that's just where I am right now. So what about this stuff? All right? Can we make biology easy to engineer? Don't know. Why not? It's different. Might be different. Maybe it is actually different. Got self-mixing molecular systems, not laying things out on a wafer. Got atomic scale thermal noise. You guys are getting to that, and gals are getting to that. These things copy themselves? No, living systems copy themselves. What's my theory for designing reproducing machines such that I can still understand the designs and I don't end up with evolutionary spaghetti code? Von Neumann didn't finish the book on that, right? The very high heterogeneity, so I'm going to have a small number of many different types of functional objects in one system. And then this last thing is a placeholder, which basically is code for if I make any incremental progress or others make any incremental progress on getting better at engineering biology, people are going to freak out. All right. Yeah. Next gen biotech via tools. So just to put this in context, this is how we're operationalizing the opportunity, if you will. The last generation has been driven in part by these core tools: recombinant DNA for cutting and pasting, polymerase chain reaction, aka PCR, for amplifying and making a few point edits, and then DNA sequencing for reading stuff out. And most of biotechnology is still operationalized on these tools. However, we're now starting to be very unapologetic about bringing in new tools that are mostly focused on design, build, test, process improvements. So synthesis of DNA, which is relatively old chemistry, but is getting improved as a process. I'll give you primers on everything on the bottom. Basically, synthesis of DNA is going to be like silicon wafer fab as a placeholder. Abstraction, which may or may not be possible to deploy here as a tool for managing complexity inside living systems. And standardization to coordinate labor. And hopefully some more ideas. And maybe one of you can figure out what that should be. Different types of coding, different types of representation of state, I don't know, different system architectures. So here are the primers on synthesis, abstraction, standardization. These are bottles of chemicals called phosphoramidites. There's a photograph taken on my desk. These chemicals are basically modified sugars. Since there's sugars and there's 4 bottles, you can infer it's ATCG. Right, so it's the basis of DNA in a form that allows for a new single strand of DNA to be assembled from scratch. The bottles cost about $250, and there's enough material, physical material in the bottles to make 30 copies of every human genome on the planet. Right, so it's quite a bit of material. The material's derived from sugar cane, right, because this is sugar. Right? So you take these bottles, you plug them into a machine which controls the dispensing of chemicals in a serial fashion, and that allows you to build a polymer of single-stranded DNA. Right? What piece of DNA gets made? Whatever piece of DNA is specified by the information coming over the computer network. Right? These machines are capable of building strands of DNA that are up to 200 bases long, 200 nucleotides long, 200 to 300, which means that if you want to build bigger stuff, you have to print out a bunch of shorter oligos and then figure out how to stitch them together. For me, this is the most important technology on the planet because it allows you to go from abstract information to physical material that, if you can bring the material back into a cell, has a chance of encoding a phenotype or a behavior in a living system. So I view this as being at least as important as silicon wafer manufacturing, and probably I'm going to say it's more important. In 2004, the biggest thing that had been assembled from scratch in terms of length was 10,000 bases long, 10,000 letters. For context, that is a small virus genome, like poliovirus. 2 years ago, the biggest thing that had been assembled from scratch was a megabase, million base pairs long. That was enough to encode a microbial genome, a circular piece of DNA. So that's a 100-fold improvement over a 6-year period. Thus, we would simply project forward in 2016, the largest thing to be assembled from scratch will be 100 megabases. That's probably technically possible. I don't know that anybody will motivate towards that length scale, except for reasons of ego. We don't have a capacity to design at this scale. We basically have a capacity to design sort of at the 10 kilobase scale back in the 2004 world. So we've got a big integration gap in genetic engineering. 100 million bases, though, is pretty interesting. It is enough for the genome of any microbe I know, Baker's yeast, for example. It is almost the full genetic complement of a worm or a fly, which are around 100 megabases across all their chromosomes. It is just under the average length of a human chromosome, which is roughly 130 megabases. So my expectation would be by the end of this decade, we're going to have a capacity to construct, not design, but construct fragments of DNA that encode most of the things we care about, if you want to devote the resources to it.
What's the limitation?
Many limitations at every level. You sort of have to approach it from the basic chemistry up to the oligosynthesis, up to the zipping up to the how do you handle big chunks of DNA without having the shear forces rip them apart. For example, a fundamental limitation with oligosynthesis is you're making a custom polymer. And so the synthetic chemistry developed by Carruthers and colleagues in Colorado circa 1982 is beautiful, but it's not perfect. So that means every time you add one base to the growing chain, there's something called the coupling efficiency. If you run your synthesizer really, really well, you'll get that coupling efficiency above 99%. But let's say it's 99%. If you're making 100 mer, what's your yield? 0.99 to the 100. What's that? 36%, I think, if I remember, right? So right there, you got an issue. How would you get around that issue? There's a really cool project in a bioengineering lab on campus. EMIG is working on it. He's trying to do closed loop synthesis at the single molecule level, where we basically use an AFM or an optical trap to sort out whether or not you've added a base. And then you just burn energy on getting higher coupling efficiencies. But at every stage in the process, right, it's an engineering challenge.
Okay, don't you want to put error correction in there somewhere?
Of course we do.
Early, not late.
He does.
The genetic code itself, for example, if you think about coding sequences, has a lot of error correction in it. Because you have a mapping from 64 codons at the triplet, for protein coding sequences at least, 64 codons to 20 amino acids. And there's been an analysis of how that mapping table is optimized to support evolution, which has some correcting, if you will, in terms of biochemical activity. I'm more interested in going in the opposite direction. I would like to see people implement fail-fast genetic codes, so that, for example, every point mutation would be selected against.
That's what I mean, basically.
Yeah, all right, fine. Well, this is just a little primer on synthesis, just tastes, right? I want to get to abstraction. Information encoded on DNA is or can be complicated, right? So TAA, TA, CGA, CTC, ACT, ATA, GGA, AGA. What's that? You'd have to memorize all these low-level sequences if you wanted to implement systems. This is typically how genetic engineering is done. People are expert in the details of every component they're using. You can memorize these sequences by having them be your login passwords. They don't have numbers in them, but they're still pretty long. So I don't know if somebody's developed a way to crack them automatically. But it really is working with low-level stuff. So very naively, I'd like to live in a world that has something that looks like this and keeps going, but I'll just give you 4 levels. Let's say I want an 8-bit synchronous counter. Please give me set reset latches or toggles. Right? Oh, please give me a bunch of, I need 16 different integrases that recognize different sequences on the DNA and no crosstalk, so I can implement them in the same self-mixing volume. And please get me the DNA. In other words, I want to live in a world where somebody could be a system designer in bioengineering and they wouldn't need to know that DNA is made up of 4 bases. Right? Or anything about phosphoramidite chemistry or how to synthesize DNA. This turns out to be technically controversial, in part because of the example I walked you through at the opening of the talk. It took us 750 attempts, about 1,000 attempts, to get a latch working. So that means I've got that many transactions across that middle interface. Right? That doesn't sound like it's going to work. Why hasn't this been made to work? Again, it could be physically impossible, but I'm going to put forward a different argument. The culture, the organization of people in biotechnology has never been such that the engineering work can be done easily. For example, we have students, they're really creative, they're under pressures to get good grades and win competitions. We have academics who are under pressure to publish papers. And the work's done by students who are under their own pressures. We have national laboratories and industry. Industry and biotechnology needs to deliver results, which are increased yields in a biomanufacturing process or a drug that makes it through trials. And the national laboratories are going to be mission-driven, overdriven. And it's not like in electronics and software where people understand that you can start a company by having strong IP and you can start an industry by giving things away. It's mostly you can start a company by having strong IP. So this is what we've got. We don't have the Universal Serial Bus Consortium. We don't have the Mozilla Software Foundation. We don't have the Apache Software Foundation. We don't have GNU Linux. We don't have blah, blah, blah. Right? So this.
It's the equivalent of Bell Labs, what it used to be.
Not yet.
That's an interesting choice of a lab too. That's a lab of physicists. That's not a lab of biologists.
It's the strongest national lab in the U.S. for biology.
I would tend to disagree.
That's my opinion.
Yeah, but you have like you have like Human Reference Genome Build 37,
Yeah.
and
Yeah.
tools like
Yeah.
genome browser. And
In a
stuff like
few
that.
cases, the biological research enterprise has gone meta. Sequencing of DNA is the best example. And note that that was not an easy thing to do, by a long shot. And I don't know of anything like that for genetic engineering.
How about this open? I don't know exactly what the time is, but the idea of do it yourself in the home.
Yep. DIY bio or bio-curious,
Yeah.
or so on. Yep. Let's come to that.
Okay.
Yeah. But it's part of this, right? Maybe we should organize. All those things on the previous slide are great. I'm not arguing against those. I'm just saying that's kind of it. So could we have new modes of organization? We tried an experiment over the last 2 years, joint between Cal, Stanford, and LBNL. Call it Biofab, which stands for Biofab International Open Facility Advancing Biotechnology, aka Biofab. So tried to be as nerdy as possible. Our dream was to develop free-to-use languages and grammars for taking the engineering of the central dogma off the table as a research question. I should be able to forward engineer a whole genome and have the molecules be expressed and controlled at exactly whatever levels I want with 2-fold resolution. All right, that's the technical dream. And then maybe we could do experiments around how we organize. Maybe we could begin to bring companies together and work on pre-competitive stuff and so on. Maybe we could hire professionals to do work in a public benefit setting. That's novel for a lot of this. So we did that. This is the work that's been led by a gentleman named Vivek Mutalik, who used to run a 600,000 liter penicillin fermenter, but then did a postdoc in gene expression control at UCSF. So he's got a nice mix, and he decided to take a risk and try and do a crazy little project like this. So what Vivek's team did was develop a microarchitecture for laying out functional elements on DNA for controlling the central dogma. So these are insulating elements, transcription, translation elements, more insulating elements. Won't unpack all the details. But the novelty here is not just the insulation, the fact that we foregrounded a consideration of what the junctions are between elements and what they might be and how to get better junctions. These rules got developed over a period of time, and then parts libraries got built out in accordance with the rules. So let me give you one example. Again, this is just brilliant work by Vivek. This is how we, in our latch work, typically handled the reading out or the printing of a protein in the cytoplasm. We have a transcription element. It's a found object. We have a translation controller. It's a found object that hasn't been regularized. And then we have a gene of interest, perhaps an integrase or an excisionase. And we slam these things together willy-nilly and hope that the level of protein made is what we want. The reason this is problematic, if you think of this as a piece of DNA, imagine that there's a piece of mRNA that gets produced. This piece of mRNA is going to have two functional elements on it. This translation controller for initiating protein synthesis and the gene. The molecule itself doesn't know what we intend. So, for example, if I have a translation controller, which is really nice and strong, it's my standard strong translation controller, it's going to have a specific sequence. AGG would be a good example. If I put an AGG sequence here, I'll get lots of translation. Unless you show up with a gene that happens to have the complement to that sequence. Let's see, if it was AGG, it'd be CCT. And if somebody shows up with a CCT gene, all of a sudden this thing will self-fold into a hairpin, and this is no longer a translation initiation element, it's a hairpin that does nothing. Right? That's an example of where we run into problems at a physical chemistry level. You're going to have to teach me that, but that sounds fine.
It's where levels cross with the unfortunate presence of the physical world.
Yeah, the physical world. Blame it on the physical world. Yeah.
Well, that's why I need the / part. It has to define itself.
Well, I don't know what that means.
Ask
So
him. He said /
Yes. Here's the solution Vivek came up with. So this is a physical architecture for translation controllers such that the element is insensitive to the downstream gene. Stated differently, the amount of translation this element produces is the same, regardless of what the downstream gene sequence is. The way this works is we still have the translation controller, but now we bury it in a leader gene, which is very short, just 11 amino acids or so long. This leader gene has a coding sequence that is nested by one base pair into the gene of interest. And what that looks like at the code level is TAA for a stop codon on the leader, and then ATG for the start codon on the gene of interest. So 5 bases, 2 codons, -1 frame shift is the term of art. Then upstream of this leader gene, we have another translation controller. And I'll show you what this looks like in terms of performance, but for now, let me just say it works. And we don't know how it works, but here's the story we like. The ribosome begins translation here, and it occupies or laminates this region of the transcript such that this second translation controller is physically busy and thus cannot fold up with anything that shows up here. Right, so we're using physical nesting and active coupling or just occupancy to decouple the function. All right, we did a / then. I don't know what that means still. It's fine. I'm very comfortable with not knowing. I'll learn later. One of the things that's really cool about this is this type of architecture is found in natural systems. But we never really understood why it was there. And as naive engineers, we wanted to rip it out all the time. And now we have, through Vivek's work, a reason to put it back. I could say more about that. But let me just show you in terms of how it works. So across the top are 24 different translation controllers. And across the rows are 12 or 15 different genes, different sequences. And what this heat map shows is, as a function of different genes, do the translation controllers behave the same? Basically, do the columns have the same color? And that's what it looks like if you use this bicystron complicated architecture. If you delete that first ribosome site, you get the salt and pepper plot here, right? So we think this is looking pretty good. Let me show it to you a different way, now in more detail, where we haven't just regularized the translation controllers, but also the transcription controllers. So across the top are the translation controllers, and across the right side are the standardized transcription controllers. I haven't shown you that junction or the architecture, but they exist. Now we're looking at the heat map for the expression of one gene, the green gene. And this is what the heat map looks like for the expression of a second gene whose sequence is 50% different, very different. And it's basically the same heat map. You will find notable outliers, for example, here to here. But it turns out we can trace these back to errors in the laboratory where there was mispipetting into the reactor well. Human error. In other words, we've now made functional composition reliable enough that we're uncovering other errors in our process. Yeah, they have even more problems than human beings. Unless you spend a lot of time professionalizing it and debugging it. But robots by themselves tend to just consume resources.
Slave labor.
Professional technicians are really great. This is work done by professional technicians in a public benefit context. That's the hack for us. Let me give you a sense of why this matters. I implied this, but here's what the data looks like. If you take found objects for controlling transcription and translation and put them together in all combination and use them to express two different proteins, red or green, and then make a scatter plot where each point represents, for a given combination of transcription and translation controllers, how much red or green protein do you get? This is what it looks like. Right, so the R squared is about 0.4. And for example, one combination of elements gives you, is log base 2, about 16 times more red than green. Or this point here represents, for this combination of elements, you get about 60 times more green than red. In other words, if you take found objects and you use them with different genes, you get almost a 100-fold swing in the abundances. I need things that have precision to factor of 2. Right? You take the Biofab produced elements, and the scatter plot looks like this. And if I put my factor of 2 window on there and slide it along the correlation, I see that about 93% of the points fall within the box. So not perfect. But what I will admit is I don't know whether or not the remaining variation is due to problems in measurement, because we don't have a reference object to remove a lot of the extrinsic factors. So this could actually be much better. But this turns out to be amazingly better than the left. And another way to say that is we now have a greater than 90% chance to hit our target windows, as opposed to previously where we basically had a 50-50 chance. So this is a crazy advance. It means we're now challenged to begin to think about designing systems 10 genes at a time. And the reason that's crazy is I don't know anybody who has a design framework that can smartly specify the levels of things you want for 10 gene systems. Right, so we've now got one problem looks like it's solved, and we have a rising of another problem.
Essentially the noise margin in the digital.
Yeah, could be.
But you still need that higher order error correction.
Probably. Although, you know, that data register we've got is looking pretty tasty.
Sure, it works as long as you do the demo. So it doesn't have to work that long,
You
right?
can also build 1 million of his and just see which ones work.
Yeah.
Without having to be so darn careful.
So I just want to cover a couple other topics, or we don't have to. This is my shopping list. This is what I would like the world to invest in. Please get better at building DNA. I want turn time worked on right now. I run the sophomore teaching laboratory in bioengineering here. We have disallowed all work and instruction on recombinant DNA. So we don't teach that anymore. We have our students design genetically encoded sensors or actuators. They have a month to come up with the sequence. We ship that to a company. The company has less than 3 business weeks to give us the intact vector back, which we then transform into the cells. When it comes back, and the students test it. Right? The reliability of doing that last fall quarter was 90%, that decoupling process, meaning professional build of up to 3,000 base pair vectors had a 90% success return in 3 business weeks. It's great. And 25% of the student encoded designs produced phenotypes that were consistent with the design that we could test in a single quarter. Today was the first day of lab, so I came right from lab. And we're running on an off quarter. If anybody wants to pop into that laboratory, we have a few more slots. So if you could get better at building DNA, this would be a big deal for us. If you could make that 3 business weeks become 3 business hours, then we would do more than 1 design build test cycle in a quarter. We'd do a design build test cycle a week, right? Rules for functional composition. I gave you an example of one rule, that bicistronic translation controller. We need a lot more of those, probably. Sensors and actuators for getting information into and out from systems. Well, yeah, I used to know what I mean by that. So we've gotten to the point where we're starting to take central dogma off the table for 10 gene systems. So one agenda would be to say, okay, let's get the reliability up to 100 gene systems. Okay, now let's get the reliability up to 1,000 gene systems. And at this point, we're at genomes, right, for microbes, and so you could march down that. I've shelved that agenda, at least for my own energy personally, because I've better appreciated that nobody has a design capacity at that scale. So I used to want that a lot, but this talk needs to be updated from my thinking over the last few months. Right? But I would like to live in a world where you could basically sit down and forward engineer a genome from a set of genetic elements that are free to use. And it would work. And we'll get to there eventually. Right? So, I still want this, but the prioritization is now de-weighted for me at least a little bit. And a different way to say that is we've got 10 gene systems working in E. coli. I now want to go horizontal across all organisms, across the kingdoms of life, and pilot 10 gene systems in any organism anybody cares about, and get those into the public domain to begin to seed what'll come later. We can talk a lot more about this. I'll just show you, this is my assessment of numerically, somewhat made up, but best I can estimate how difficult this will be to do technically, sort of where the sticky stuff is and where the easier things are to do. The functional composition hacks are really crazy. Sensors that allow you to transduce chemicals into signals have really lagged. There's some work going on here and elsewhere, but it's tricky. So we come back to this, but let me cover a couple other topics. So when we started working on this type of stuff, meaning stuff is process improvements, our audience was students. And for example, this is a course we organized back east. You can see Tom just off center in the right, then Randy Redberg, and then Jerry Sussman, 2 in from the right. We taught this in the old AI lab. And these students tried to implement genetically encoded oscillators, getting to the DNA designs in 1 month, and then we were going to ship the information for synthesis and get the DNA back, and they'll be great. We got the designs done in a month. We were 50% over budget. I had $80,000 from DARPA to pay for the DNA synthesis. We had to figure out how to share parts so we could reuse components to cut the cost of our synthesis order. That gave birth to something called a registry of parts, which is online, partsregistry.org. Our synthesis order then got to $67,000. I placed that order. Nothing came back because the student parts crashed the build process because nobody had figured out the rules and back promulgated them to the designers. We worked with the companies. It took us 7 months. We got 58 of the 60 parts we ordered back. We didn't have time to test anything because the next course was coming up. So the insight was, don't worry about that. We've learned enough. Teach the course again. So we did. Now we're trying to do program pattern formation based on people like Radhika Nagpal's work and so on. And we ordered all the DNA right away. It all came back within a month, and none of it worked in terms of the systems performing. That was so great that we taught the course again that summer. We didn't even wait a year, and we went to 5 schools. And one of the projects ended up working. I'll show you this later. And that was so great that it went to 13 schools and then 39 schools, and a world map came out detailing what was then called the iGEM competition and where all the teams were coming from. International Genetically Engineered Machines Competition. So genetic engineering Olympics, if you will, for college undergraduates. Where at the beginning of the summer, they get a collection of parts, which is all the parts made by the previous year's teams. They work on a project of their own spec, and then they show up at the end of the summer or early fall and present and celebrate and compete. We brought China on, meaning the students went over there and recruited those schools, and they then hosted us for a weekend workshop. They won the next year. The team from Peking University won, and it got a little bit bigger and a little bit bigger. Randy Redberg, who had retired from the Sun Microsystems storage office, has taken ownership of this and is running it and scaling it. It got a little bit bigger. Here's an example of what these teams are doing. This is a project from Cambridge, England in 2009 called Escherichia chromai. Reprogram the metabolism of E. coli to produce pigments, the 7 different pigments shown across the top. I wish they'd call it E. Crayola or something like that. They pulled it off. 16 weeks, 8 teenagers, $25,000 in capital, implement 7 biosynthetic pathways. How did they do that? Half of the genetic components needed to do this were in the kit they got at the beginning. A DNA synthesis company that happened to be founded by a graduate of Cambridge gave them free unlimited DNA synthesis. And they got a chassis, a strain that overexpresses some of the precursors to make the carotenoid chemicals on the left from DuPont. You put all those things together, they did that. What do you do with this? What's the miracle application of E. chromai? Don't know. They're actuators. They got together with the Royal College of Art in London. The RCA folks came up with this. The Scatalogue. You have a probiotic that has sensors which detect the state of your alimentary canal, and depending on how you're doing, what comes out of you is colored, depending on the specialist you should consider. All right, so this example of getting information out from a human system into a easily readable form. That's quite valuable. Better than colonoscopy. Here's another project that got things going back in 2004.
Was that published? Was this published?
It's on the internet.
It's all over the place.
Is anybody actually trying to
Commercialize this?
yeah, or implement it or use it?
Not that I know of. We don't have a consumer biologics market. Well, we'll come back. I know we're running out of time, so I just want to give you some snapshots to think about. Here's another project. This is a sensor, adding a photoreceptor to E. coli, spread the E. coli out on a plate, hit them with light, they take a photograph. All right? This is published in a easily found form. This is a project done by high school students up in San Francisco at UCSF for the summer. They were inspired by Java, the programming language. So they started their project by watching the videos on YouTube of past teams and doing a failure analysis. And about half of the teams that didn't get something working had the lame excuse of, I just couldn't get the DNA I wanted in time. That's why I want DNA Fab. They couldn't do anything about that, they thought. But the second most prominent excuse was, we put the DNA into the cell and it didn't behave. Biology's complicated. They thought they could do something about that because that's a really lame excuse. And so their idea, based on Java, was to say, well, what if you just didn't toss the DNA into a cell willy-nilly? What if you installed your designer DNA in a virtual machine, in a well-designed sandbox that provides a common operating environment such that you know what you're designing for? And if we invested in that, then maybe we'd have more reliability in getting things to work. And oh, by the way, if you got such a virtual machine working in Saccharomyces cerevisiae, maybe you could get it working in the same one working in people and plants, and so you'd have real nice horizontal portability of your code. How do you do this? They decided to try and build new organelles, compartments inside cells. Their idea was to redirect the invagination of membranes that lead to the budding off of little membrane vesicles in the cytoplasm, accumulate those, then modify the phospholipids that allow things to be targeted to them specifically with a biochemical addressing system. These are high school students, right? They're teaching me this biochemistry as they're giving their presentation. And they prototype in yeast the beginnings of what looks to be the accumulation of membrane vesicles in the cytoplasm of yeast that can be targeted with a fluorescent protein recognizing a phospholipid brought in from a human. Not bad for summer. Right?
Isn't that what you need for error detection?
I don't know. Yes. Okay, yes. I don't know. All right.
First stage.
So Some other things come to attention when you make such incremental progress. I mentioned this before, let me unpack it. Who's going to lead biosafety into the future? We have a great biosafety framework that was pioneered by Paul Berg and others here from Stanford in 1975. It's not perfect, but it's an operational success to a first approximation. Some can disagree. But we now need a renewal of leadership, not just practice, but leadership. Second one, what's our strategy for biosecurity in a world where we get incrementally better at engineering biology? I'm just going to say incrementally better. But how much can we make with biology? Are we going to manufacture all our energy? What's the best balance between private ownership and public ownership? Who's choosing what gets worked on? How do we not have this be merely the industrialization of biology based on old modes of engineering? Shouldn't we expect to learn new ways of doing stuff? New ways of error correction? I don't know. How can we best work together? So there are more questions, but I'll just open with these and then give you my self-assessment of how difficult it'll be to come up with solutions. For example, I think the stickiest one is biosecurity right now.
Separation.
Plan for dealing with intentional misapplication of biological technology. Who's got the plan? Who owns the plan? Who's being held accountable for implementing the plan? It probably isn't just one person or one organization, probably very interesting structure. All right, here's the last thing. The different project I've been involved with is called Synthetic Aesthetics, where we brought together creative designers and artists with would-be engineers of biology, and we paid for them to spend 1 month in the lab and 1 month in the studio. And our motivation here was to explore new applications of biotechnology that aren't just about bulk manufacturing of chemicals and drugs and stuff like that. So I'll give you, you can learn more from this website. Let me give you one quick vignette. So one of the pairings was Sissel Talas, an artist from Berlin, and she worked with Christina Agapakis, a molecular biologist at Harvard. Sissel arrives in the lab at Harvard and asks, what's interesting in microbiology? Well, what was interesting that day is reports on how people have been sequencing microbes that live on human skin. Things on the outside of your elbow are alike and different from the inside of your elbow. Right? What are you excited about in the world of art? I'm interested in the smells of cheese, says Sissel. Oh, we have a nice cheese shop near Harvard. Let's go and get a special tour of their aging cave, and we'll bring a rack of test tubes and we'll get core samples from the stinkiest cheeses. And then we'll bring these cheeses back to the lab and streak them out on plates to begin to study the microbes that make the cheese. Okay, great. Now some of these small batch cheeses, they realize, are made by human beings manually. What's the relationship, they ask, between the microbes that live on our skin and the microbes that make our food, cheese in this case? A classical approach, based on a scientific method, would be to start sequencing and studying the microbes from each and doing alignments and comparative mappings. But in engineering, a synthetic approach would be, which is what they took, let's go around Harvard and collect microbe samples from the skins of our colleagues. And let's go to the farm and get raw goat milk. And then we'll bring that back to the lab, inoculate 48 different bottles, and make cheese. So they do that. And I happened to be going through Boston at just the right moment, and they came out with a platter of 48 different cheeses, including cheese made from the microbes collected from Daisy's armpit, from the nose of Sissel, from the toe of a philosopher, and so on. Daisy's armpit cheese turns out to be beautiful. It has, it's a stunning, I'll come to that. It's a stunning citrus floral bouquet. We just smelled this. We didn't taste it because of biosafety. Hadn't figured that out. Philosopher toe cheese, you need a new adjective to describe how bad it is. Right? So wrongest was the only word I could come up with. So I thought this was really, really, really just amazing. It punched through something that had been separated for me, right? My food supply and me, we don't relate. But this basically trumps what my parents told me, right? Instead of this, you eat what you are, right? That's kind of interesting. When I had dinner that evening with a banker and thought this would ruin his appetite, instead he immediately replied, $billion-dollar market. Celebrity cheese. Celebrity cheese. Right? Barack Obama blue. Right?
Yeah, but you might have to go for the most stinky one.
There's apparently a startup called Sandhill Foods. Not associated with them, but they could do something like this. So if you imagine what biology can make, right, it's very different from how it's been classically imagined. The skeleton of a sponge, for example, grown in place in the Pacific Ocean. This is a 10-centimeter scale. This work was done originally at Bell Labs because they were interested in the optical properties of the skeleton. This thing self-assembles itself in the environment. Pretty crazy. A suspension bridge, again, grown in place from the roots of a rubber tree. It fixes carbon over time. It gets stronger, right? Pretty interesting manufacturing capacity in biology. This is a fantastic fiction out of the architecture program back east where people were imagining growing houses, gigantic programmable gourds that differentiate into 4-bedroom, 2 baths. Note that the physical substrate manufactures at this scale, right? Okay, so I have zoomed out now all the way, I hope. We're facing a challenge in bioengineering, which is, can we make biology easier to engineer? I think the meta lesson, which we're really struggling to implement, is how to sustain investments in tools. This appears to be, meaning this region, the only place on the planet where people tend to get this. So I hope that part of the future renewal of whatever is called Silicon Valley might more explicitly encompass tools infrastructure supporting engineering on carbon, nitrogen, phosphorus, oxygen, sulfur. The worst case scenario in terms of knowledge is we can't deliver anything, but we learn a lot more about how biology works by tinkering as a complementary approach. Opportunities are to pull off basically a remaking of aspects of our civilization associated with manufacturing. And that should be good so long as we manage some of the tricks. So I want to thank too many people to say out loud, Jerome and Tone in particular here at Stanford for their excellent work on the data storage register, Vivek Mutalik and the colleagues up in Emeryville working on the Biofab team, and approximately, 100 to 500 engineers and scientists who have allowed me to do a crazy exploration of different types of research. Thank you again for the invitation, your patience. Happy to stick around. Whatever you would like.
We have time for a few questions. Okay. So you raise your hand.
So how's funding in this area?
You talk to the bank or?
The funding, how is funding in this area? If I talk about tools work or?
No, I'm looking for the sort of unconstrained. I was around in the early ARPANET days, and one of the interesting quirks was that Doctor sort of pushed you in the right direction, but if you did something else, that was okay. You know, email was a hack that Tomlinson did one night. So that's the sort of funding I'm looking for.
So
That went away.
this is the study I briefed out to the director of DARPA in October of 2003. Okay, and care of ISAT. Ed Lazowska was the chair of it at the time, and basically, what it, can I make it bigger? Yeah. Basically, what it said is we wanted to change the engineering process from one-off ad hoc to scalable. And it outlined how to do that, and you'll recognize some of the language: standardization, abstraction, and decoupling of design and fab. So basically that bottom row. This brief went over like a lead balloon, and in part because of the scrutiny associated with total information awareness, and because nobody has a security plan for biotech. And the political exposure around that can be significant. Almost 10 years later, a hero back in Washington by the name of Alicia Jackson has initiated the piloting of a very small program called Living Foundries out of, not DSO, I think it's MTO. Okay, that's as close as we've got to something that's an unapologetic tools investment. It is underfunded and brutally overdriven in terms of timing of deliverables. It probably needs to be about a factor of 10 bigger. We're also running into an issue, which is it probably is not strategic to have the core infrastructure for the future of biotech to be developed only with the DoD funding. We need to have a very significant scale up and renewal from NSF, I believe. And we also need to see NIST get into the game at even a modest level. So I would say the public funding landscape is very fragile, speckled, and still very early. The largest public funder of this work at the moment would appear to be China in the form of 4 key laboratories in synthetic biology that were brought online, whatever that means. I can say more about that, over last year. And they're a cash-rich research environment right now. When researchers there, who are my colleagues, put in grants, they get asked to ask for more money. Having said that, I think our colleagues in China are under very strong constraints with solving immediate challenges with respect to environment, water, food, energy. Right? So how they'll do in terms of delivering tools infrastructure and innovations at the tools level, I don't know yet.
You said you could say more on this subject. Say more.
In what direction?
About those labs. You said you could say more about the labs in China.
Well, a good example would be the BGI in Shenzhen, the Beijing Genomics Institute. This is probably the largest DNA sequencing facility on the planet. They operate in a converted 9-story shoe factory. They employ of order 4,000 people on an annual budget of 120 million US, so far as I know, roughly. And they're the largest sequencers of DNA. They've decided to initiate a new synthetic biology unit, right? And the conversation there is, what fraction of our workforce should be devoted to synthesis? 1% or 10%? And the last internal discussion I saw was 10%. This is important. Sequencing and synthesis are intimately related, and we got a lead in this. The average age at that facility is 25 to 30, somewhere in that ballpark. It's run by a 35-year-old. What will they do? I don't know.
So is there a emerging venture community for this? And is it, are we going to begin to see products?
There have been investments in tools. They have not been hugely successful, and they have not sustained. A lot of the investments in tools end up being investments in sales and marketing around tools, and not actual investments in technology improvement. We're still working on this. So I'm, for example, in the middle of a second venture-funded company to get better at building DNA. And the first one crashed and burned because it didn't manage to spend sustained money on technology development. It got so good at sales and marketing that the factory got overwhelmed with orders, but it had no capacity to deliver against those orders, so it blew up. The second one is, yeah. And that organization is doing exactly the opposite. It's not on the market with anything to a first approximation, and it's 3 years into tech development. And the question there is, how do we sustain funding for tech development? I don't know.
Okay.
Yes, sir.
Slightly different question. Could you give a sense of what a programming language for some of these biosystems would look like? For instance, your hairpinning example, I'd imagine that a programming language with a really strong type system might address some of the problems that you had there.
I think it would depend on what level in the stack you want to be coding, right? So, for example, down at the primary sequence level, you would have a lot of rules you could begin to implement as checkers, right, to find conflicts. You could begin to kick out to computational methods for predicting performance, right, that would begin to push information up to a functional object level. So, for example, how strong is this translation control element likely to be in this specific context? There are ribosome binding site calculators that allow you to estimate that. So you could go from primary sequence up to functional component. Once you get up to functional component and to the device level, I think languages become relatively familiar within the context of laying out the types of systems I've hinted at here. If growing, print isoamyl acetate. If resting, print methyl salicylate. That would give you a cell that when it's growing smells like bananas, and when it's in stationary phase smells like wintergreen, right? So at that device to part level, you'd start to see stuff like that. What I'm most excited about as a naive consumer of programming languages is when you keep going up and you start to envision things like an autonomously generating bridge or an autonomously generating limb or a house. And I now need 4-dimensional programming languages to program in time and space. So as a naive student of things like amorphous computing, right, I find the invention of and playfulness of languages in 4D contexts interesting. So there's a lot more we could explore there, but I guess my placeholder would be if you think about an emerging abstraction stack, and oh, by the way, the significance of the Biofab work product is we've now demonstrated how you can begin to get success across the device to component level, right? And that's the first example of that. So I think that abstraction stack, which has been technically controversial, is going to blossom because I think it's physically plausible. So low-level checkers, we need to figure out what the device families are and how to represent what we care about in whatever physical way we want, and then we can talk about languages maybe, I guess. And then once you get above that, it touches upon things you're probably familiar with, and then I'd look to 4-dimensional programming capacity. One last thing, sorry. Biological systems operate on what I think of as called 3-time. So they have to perform on 3 time scales. The immediate time scale, the time scale of learning, and the time scale of evolving. All right, so for example, how do I get a signaling pathway to just transduce a signal? How do I get a signaling pathway to adapt its response as the concentrations change by a factor of 1,000,000 outside the cell? And then how do I get a signaling pathway to evolve or not evolve depending on what the performance spec is? Right, it might be needed, for example, once every 100 generations, and I don't want it to atrophy in the absence of positive selection. Right, so another interesting element of programming and design is when you think about solving these problems, it could be the case that the solutions to design in each of the times gets mapped in part onto the same architecture. So there could be a really interesting way of representing that from a programming perspective. Right now, when we look at natural systems, I suspect one of the reasons we see so much apparent complexity is we see these 3-time solutions overlaid onto a single instance, and how do you deconvolute that?
That's it. Best to be able to build it once in your bench.
Yeah.
But there's a whole other layer of design constraints that
Yeah.
you face if you want to be able to mass produce it reliably.
Yeah.
And then there are yet additional constraints if you want to be able to build a business around it.
Yeah.
And sell them
Yeah.
and have consumers
Yeah.
appreciate them, demand them, and all
The one that it might be different is your phone you're holding doesn't have to copy itself.
that.
But if it did, it would be really nice.
Yeah.
Or just a new copy.
Yeah.
So, I have an observation, it's feedback from listening to you today. And it sounds to me like the analogy for kind of where you are and where you want to go in a short period of time, like 5 years, is TTL logic.
Yeah. Great. So, Engineering the World, celebrating the 75th anniversary of TI. Amazing book. I studied it because I wanted to know, how did they pay for the TTL data book? And I came to the following conclusion. I need to militarize biotech. And I immediately rejected that conclusion. So give me a...
Chinese already do that?
Give me... No, I hope not.
Are you sure?
No, I hope not. How do I establish trust across barriers of secrecy for biosecurity? Right? The whole other lecture. And I don't know where it ends up. So tell me how to pay for the investments in engineering infrastructure in a space where I don't have a consumer biologics that supports fast turn. Instead, I've got monolithic, gigantic one-off projects where most of the capital and time costs are downstream around safety testing. And I don't want to militarize or remilitarize. Right, so I need the economic hack. Consumer biologics is a big deal if we could figure out how to pull it off. Or other sectors, right, could be totally different in this space.
Well, thank you very much. I'm sure that there are other people here who have questions. They can come down and talk now that the tape is off. Thanks very much.
For more, please visit us at stanford.edu.